Hello.
Hi.
Yeah, can you guys hear me?
Yes, we can hear you. You're audible.
Okay, okay. So I'll wait for 5 minutes more, and then we can start.
Sure.
Hello.
Yes.
Uh, yeah, since no other people are joining, so hi, I am Nilanjana. I am a PMRF scholar and I am a TA, teaching assistant in this course of Professor Himanshu Kumar. So, I will ask you whether you are having any doubt in this week's course.
Well, exactly to say not that much, but then no, since it was an interactive session that has been suggested, so I thought maybe to come and see what has been going on here. Even though this is my first time taking classes online, I mean attending classes online and so on, so it will really be a great pleasure.
Okay, okay. So, so from where you are?
I'm from South Sudan, but I'm doing my BSc in Chennai, Advanced Zoology and Biotechnology in Loyola College.
Okay, in biotechnology. Okay,
Yeah.
okay. So you are interested in immunology?
Well, I have interest, a really great interest in immunology, though I find it a little bit complex at some point. So, since I'm planning to pursue my PhD in virology. So
Okay,
it's
you
like
want
kind
to
of
pursue
research.
your PhD in virology.
Exactly. Yeah.
Okay. So, s- the like why you want to pursue your PhD in virology? Like,
For me, it's interest. Human body is one thing that is I found it is really complex to understand. But then in my interest, I want to understand a much deeper knowledge on how virus come and how it affects our body. Because for me, the pandemic has been a great lesson for me, and then I think it has been an eye opener for me because I was like planning to look for courses to join after I finish my senior four, and then I think virology, I think is for me. I found a path there to work on.
okay, okay. So, what do you know about viruses currently?
Well, not that much really, but then I'm still trying to figure out to say, yeah, because I know viruses they cannot survive on their own. They need a host to survive on, and then
Yes.
viruses they differ according to sizes. Yeah,
So, are
and
they living entity or
no, they are not,
virus?
since they require a living entity to survive on.
Yes. So, I personally work on RNA viruses. My PhD is about RNA viruses. I especially work on influenza and dengue virus.
Wow, I see. So at least we have something in common.
Yeah.
Mhm.
Uh, uh, Hello. Uh, Someone has done Shilpi, Dr. Shilpi.
Yes, yes, ma'am.
Uh-huh. So, do you have any doubts regarding this week discussion?
No, ma'am. Actually, I have just joined to see whether there is a problem solving activities there. I think that's why I have no doubt right now. So,
Okay.
is it sure that if we have doubt, then only we have to join, or
No, no, no. It is like if someone is having any doubt or so I am asking whether anyone is having any doubt or not so that I can clear it.
okay, okay, ma'am. Okay.
Okay. So has uh has everybody listened to this week's talk?
No, ma'am. Actually, second week lecture is remaining because I'm doing job right now. That's why it's very difficult to listen with the lectures only. So uh, I'm trying to listen all the lectures at the night only. That's why second week
Okay.
lectures is remaining still now. Yes,
Okay, okay.
yes.
So hi everyone. I am Nilanjana. I am a teaching assistant. I am doing my PhD in RNA viruses and host immunity under Professor Himanshu Kumar only. So currently, I am a PMRF scholar, and so if this session is all means if anyone is having any doubt regarding this week's course, you can tell me. I have also prepared a presentation for all of you guys regarding whatever sir taught. Regarding that only, I have made some presentation which I can teach you again. And if you're having any doubt in that, you can ask me also again.
Okay, okay, ma'am.
Uh, is my presentation visible to everyone?
Yes, is it? It is just now. It's only screen your home
Okay,
screen of Google Meet only.
okay, 1 minute. Is my presentation visible?
Yes, it is.
Okay. When did someone wants to join? Uh, Hello everyone. So I'm Nilanjana. So I'll be teaching a brief about whatever Sir has taught in his first week. Some more information I will give. So Sir has taught about the history of immunology. So I will briefly talk about the overview of the immune system currently in this week one lecture. So here you can see like this is a beautiful picture of a human macrophage that is colored in red and which is ingesting a Mycobacterium tuberculosis. You all know like what is Mycobacterium tuberculosis. That is the bacteria that causes tuberculosis. So first, as Sir has already taught a lot about the historical perspective, so can anyone tell like uh, who was the first one to actually report about this immunity? Can anyone in the group can tell? No one? Okay, so first the it was early in 430 BC that this great historian Thucydides, who was the first one to state that there is something called immunity. That is, immunity is a state of protection from infectious disease. He was the first one who has written during a plague was going on that time in that place, and he has written in his some book that there is something called immunity, which has come from the term immunis, which is a Latin term meaning exempt. And he has written that those who had recovered from the plague could nurse the sick because they could not contract the disease a second time. So this was the first time like a person has written about immunology during that 430 BC when there was no knowledge about our like whole physiology. So this was the first time that he has stated about immunity. So we all know what is vaccination. So can anyone tell here what is vaccination? Anyone in the group? Alias?
Yes, ma'am.
You told you wanted to study about virology, so what is vaccination?
I think in a simple term, I can say they are biological boosters that improve the immune system against a certain antigen.
Yes. So, so vax. So first, early vaccination studies led the way to immunology. So what happened? It was the first recorded attempt to actually induce immunity were performed by the Chinese and the Turks in the 15th century. So that much earlier, this first attempt to actually vaccinate people came. When it came, it was a smallpox outbreak. So smallpox is a disease which is caused by a RNA virus. But currently, we know that there is no smallpox. We are not hearing any smallpox cases around us because this vaccination has eradicated smallpox already. So this early vaccination studies already started with the smallpox. So what happened? This smallpox people were having these pustules. Means here in the picture we can see this boy, this African child with having this rash typical of smallpox on his whole body. And this smallpox is caused by a virus which is variola major and has a 30% mortality rate. It is very harmful. Like people can die due to this smallpox. And survivors are often left with disfiguring scars. So, what in the 15th century, what people did to save people from smallpox was there were dried crust from the pustules or this whatever is there in the body of the person from that pustule. They have taken some dried skin or some fluid and have inserted into the small cuts in the skin of the healthy people, and they saw that the people with whom they have put this fluid or this dried skin are safe from the smallpox. So in 1718, there was one wife of a British ambassador, Lady Mary Wortley Montagu, who observed these positive effects of variolation on the native Turkish population, and she performed this on her own children. So, what is variolation? This variolation is like putting from a infected person, you can inoculate that infection into a healthy person to so that the healthy person do not again contract the disease. So, so we all know that Edward Jenner is known as the father of immunology. He was an English physician, and why he is called the father of immunology? Because he has done so much. He has so much contribution in like curing this smallpox thing. Like in 1798, he saw that there were milkmaids who had contracted the mild cowpox disease. However, these milkmaids were not having any reaction when they are uh, coming in contact with the smallpox patients. So what he observed that these milkmaids were completely immune to the smallpox because they already had the cowpox and they are immune to smallpox. So what he did, he took an 8-year-old boy with fluid from a cowpox pustule and he infected the child with smallpox. As predicted, however, the child did not develop the smallpox. So uh, this was the, as I said, the smallpox is already eradicated from the world. And the last known case of naturally acquired smallpox was seen in Somalia. So, with the help of the vaccine, first, which was done by Edward Jenner, this smallpox was eradicated. So here we can see a pictorial representation like this Sarah Nels, who was a milkmaid in Edward's place, and he saw that this Sarah Nels was infected with cowpox. And this James Phipps is what he did was he inoculated the cowpox pus from Nels, and he saw that Phillips fall ill with a mild case of cowpox. However, it was not intensified, means it is a mild disease only. Then what he did, he took from a smallpox patient again, he took some scabs or that pustule or dried skin he took, and then he inoculated Phipps with the scabs of smallpox. Then he saw that Phipps is unaffected, so protection is complete. So first what he did, he first inoculated the cowpox pus from a cowpox patient. Then he inoculated this boy with the like with some dried skin from a smallpox patient. Then he saw that Phipps was unaffected unaffected, and there is no case of smallpox in Phipps, though he was inoculated with from an infected patient. So, next major advance in immunology was the induction of immunity to cholera. So, we all know Louis Pasteur, who is known as the father of microbiology. So, he discovered that a weakened or attenuated strain could be administered to people to provide immunity against a disease. So, he termed this attenuated strain vaccine from the Latin vaxa, meaning cow. So, it was done by Louis Pasteur, like in a small village of Pouilly-le-Fort in 1881. Pasteur first vaccinated one group of sheep with anthrax bacteria. Anthrax bacteria is, or Bacillus anthracis, it was. What happened was, there was a story like Louis Pasteur once what he did was like he isolated the bacteria from a chicken who was infected with this anthracis, and he by mistake inoculated the bacteria and forgot about that bacteria, and he went to holiday. After coming from holiday, he saw that the bacteria is still there. So he inoculated that bacteria in some healthy chickens, and then he found out that when these healthy chickens who were inoculated with this bacteria are again infected with this bacillus, they were not getting that disease. So in that manner, he found out that if we attenuate or if we destroy that, like heat inactivate a particular bacteria, and if we put it into another person, then it can provide immunity against that particular disease. So he in 1885, in 1881, he again did this with Bacillus anthracis in sheep and saw that similar to the chickens, these sheep were also immune to this bacteria. And in 1885, he administered his first vaccine to a human. Here we can see Joseph Meister was the human whom he first administered this vaccine, that is the rabies vaccine. That is rabies. Anyone knows what is rabies? In the Anyone can unmute themselves and tell. because this is an interactive session. So anyone can tell any confusion or any doubt they are having. So what is rabies? Okay. So anyone, no one knows what is rabies.
Mom, it's let's say it's like when a dog bites a person, I think it goes and it affects, it damages the brain tissues, and then
Yes.
the symptoms are usually dehydration. The person will not be able to take water, so it's like
Yes. Hydrophobia. Yes.
yeah, exactly.
Yes. So Louis Pasteur administered this attenuated rabies into a human being who is Joseph Meister, who was just bitten by a rabid dog. And he administered it fast because if this rabies is going into the brain, then there is no chance of that person to survive. So before that, he actually administered this rabies virus into this human, and then he saw that he survived. And interestingly, this Joseph Meister also became the caretaker of the Pasteur Institute later. So the principle behind vaccination is that exposure of an infectious agent can result in immunity to a more or real dangerous infectious agent. So goal of vaccination, as we all know, is to expose the individual to a pathogen or a fragment of pathogen which can be heat inactivated in a safe way, allowing our immune cells to respond and developing a strategy to find this pathogen or others that are similar, and so that this pathogen can actually able to produce extremely specific and long-lived memory cells which can protect the host from the pathogen for many decades. It should be not a short-term effect, it should be a long-term effect. So has anyone heard about the term herd immunity? Anyone here knows what is herd immunity? Uh, anyone can tell anything.
It's like injecting the person with the whole virus so that they develop the immunity while the virus is within the system.
Sorry, can you repeat, Elias?
Inserting the virus within the body system of so that at least the person can develop immunity and resistance towards the virus.
No. Herd immunity is something. Uh, can you see the screen?
Yes.
Huh. So herd immunity is, suppose we live in a community. So in community, suppose if only a few people, suppose in case of COVID is going on. So if out of in a community 100 people are there. So out of those 100 people, suppose only 5 people are vaccinated and 95 people are not vaccinated. So what happens? That 95 people are unvaccinated means the disease will spread very fast. But if out of 100 people, 95 are vaccinated and 5 are not vaccinated, that disease won't be able to spread. So this is known as herd immunity. When most of the people in a community are vaccinated, that particular population becomes safe to a particular disease. Because if suppose in 100%, if 5 are only like having vaccination and 95 is did not get vaccination. So what happens? That 95 can transmit this disease among one another. But if 95 is having have got the vaccine, then that 95 are safe from the disease. But that five people who can also transmit among each other, but it won't get spread so much as the other population. So this is a herd immunity. So herd means a group of people who are protected from the disease due to vaccination of most of the people. So here we can see that there are different types of infectious diseases before and after the introduction of effective vaccines. So smallpox, as I told, the smallpox has been eradicated by vaccination. If all the people here are vaccinated, then there is no chance that smallpox can affect anyone. Similarly, diphtheria also we can see pre-vaccine how many cases were there, and post-vaccine there is eradication of diphtheria also. And similar measles, mumps, pertussis, these also have almost reduced by 98 to 99%. This paralytic polio, rubella, all this has been eradicated just by vaccination. And tetanus and invasive Haemophilus influenzae, which is a bacteria, both of these are also eradicated after vaccination almost. So pre-vaccine era and post-vaccine era, we can see that how much it was such a big contribution by Edward Jenner and Louis Pasteur. Both of them has contributed so much in discovering this the basis of this vaccine, that many diseases which has caused so many deaths have actually been eradicated. So, uh, uh, hello. So, anyone is having any doubt regarding whatever I have taught from till now onwards? Okay, so no doubt. So, as Sir has already taught about that immunity involves both humoral as well as cellular components. So, can anyone tell like what are the humoral and what are the cellular components of our body, of our immune system? Uh, Anyone can tell. This is just a discussion thing which we are doing. So, it need not be only I am speaking. Everyone can speak. Uh, Sneha. Okay. Elias, you can tell what are the.
Well,
Hello.
I guess cellular immunity is based on the cell level, and then humoral immunity can it be like um adaptive one? Can it be like adaptive one maybe?
Adaptive means what? What are the components?
I think that when now there it comes to the organs, moms, the organs that helps in immunity.
Yeah.
You have the
Organs that
thymus,
comes
the
in.
spleen, and
Haha, so
yeah,
tell.
I guess yeah.
Organs. Okay, not organs. Humoral is B cell. Jobin, you can speak. Humoral is B cell mediated. Yes, it is true. Humoral is B cell mediated. So we currently know that this vaccination is working. Pasteur showed, Edward Jenner showed that if we actually put some, like from a diseased individual, if we are putting any uh, or like thing from a diseased individual to a healthy individual, it is doing some sort of protection to the normal healthy individual, and then they showed that vaccination worked, but nobody knew that time that how it worked, what are the components in our body which are actually making this work. So in 1883, Eli Mechnikov demonstrated that cells also contribute to the immune state of an animal. So he observed that there are certain white blood cells which he termed as phagocytes, which can actually phagocytose microorganisms and other foreign material. So, and apart from him, there were von Behring and Kitasato who demonstrated that apart from the cellular component, there are liquid or non-cellular component also, which is actually helping in this immunity. So there were two thoughts at that time. One group of people were telling that there are cellular components that is contributing to the immune state of an animal, and another group was telling that there are non-cellular components which is contributing to this immune system. So here we can see that these white blood cells which are actually surrounding a foreign particle by Eli Mechnikov, this was a drawing. So he showed that these white blood cells, which he termed as phagocytes, this ingested the microorganisms and other foreign materials. This was in 1883, and here you can see that this modern image, that how technology has developed, and we can actually see under microscope a phagocyte which is engulfing a bacteria that is a Mycobacterium tuberculosis, and how it is engulfing that bacteria. So there are two types, like the cellular and the humoral components of immunity. So humor means fluid. So this humor or this fluid are the soluble component which can neutralize or precipitate toxins and can agglutinate, means clump the bacteria, and can kill the bacteria. So this component was named for the activity it exhibited, like antitoxin, precipitin, and agglutinin. It acts as an antitoxin. It can go and clump, it can go and precipitate, it can interact with the particular toxin. And so this name came because it is present in our body fluid, the soluble component. So it is known as humoral immunity. So during the 1930s, mainly through the efforts of another scientist, Elvin Cabot, he showed that a fraction of serum called gamma globulin, now we know call this as immunoglobulin, was responsible for all these activities. So these soluble active molecules in this immunoglobulin fraction are now what we call as antibodies. So these antibodies were contained, as I already told, human body humor or body fluid. As these were contained in our body fluids, these immunologic events they participated in, it was known as humoral immunity. And antiserum, that is the serum from the antibody containing serum fraction from a pathogen exposed individual derived in the case from horses, was also given to patients suffering from diphtheria and tetanus. So this antibody was discovered during 1930s. So passive antibodies and the So what are passive immunity and what are active immunity? Can anyone tell what are passive immunity and what is active immunity? Anyone can answer what is passive immunity and what is active immunity? Anyone? You can also write if you don't want to speak, you can also write in the message as Jubin has written. Aishwarya, Elias, Ariraj, Jubin, Sneha, anyone? What is passive and what is active immunity? Active immunity, which is produced by the production of antibiotics. No, Sneha, it is not correct. So, active immunity is the, sorry ma'am, antibodies. No, this active and passive immunization are both caused by antibodies only. But what is active and what is passive? So, active immunity is what is already present in us. Like we have our own immunity means that is active immunity, and passive antibody or passive immunity means we are taking from a different individual. It is not present to us by birth. So whatever vaccine we are taking, whatever other antibodies we are taking from a other person, it is giving us a passive immunity. It is not present in us from the birth. So here you can see the experiment by von Behring and Kito Sato. Like what they did, they took healthy rabbit and they inoculated this healthy rabbit with diphtheria toxoid, and they that means they have vaccinated the rabbit. So this rabbit became resistant to diphtheria. So what they did, they isolated the serum from this diphtheria resistant rabbit and they inoculated this healthy rabbit with this serum, and they saw that the rabbit is living after they treated this with virulent diphtheria. But in case also they took healthy rabbit where they have not inoculated this diphtheria toxoid, and they saw that the healthy rabbit died. So from here they found out that these protective substances were present in the serum because they isolated the serum from the diphtheria resistant rabbit. And this serum only, when they transferred into a healthy rabbit and later on they put diphtheria into that healthy rabbit, they found that that rabbit lives. So they found out that these protective substances might is present in the serum. So they found out that immunity can be passively acquired, and there are certain substances which are present in serum which can give this immunity to healthy people. So this was another groundbreaking experiment by von Behring and Kitasato, which actually is the basis which laid the foundation that these antibodies are present in the serum and the serum can infer immunity to healthy patients. So another story was, here we can see like Leonard Seppala. He was a Norwegian. There were in 1925 there were diphtheria in Alaska. There is a place known as Nome where there was a outbreak of diphtheria. And what happened? There were no antibody of diphtheria present in that place. So people were dying in that place. So what he did, Leonard Sepala, he led a team of sled dogs. Like you know that dogs in the cold countries there will be sled dogs, and from in this map you can see from Willow to Nome, he actually led this team of sled dogs and took antibodies from this Willow place and actually saved the life of many people in Nome. So this is known as a currently Iditarod Trade Sled Dog Race, because to that time this person has saved so many lives by taking the antibodies from a distant place to Nome. They to commemorate his memory, they now lead a sled dog race from this Willow to Nome to commemorate this historic delivery of a life-saving antibody. So this was about humoral immunity, like the antibodies. Then it came about the cell-mediated immunity. So there were different people who actually showed that apart from this humoral immunity, there is another branch of immunity, that is the cell-mediated immunity. So in cell-mediated immunity, it was a key experiment was done in 1940 by Medill Chase, who was working at the Rockefeller Institute. What was done? He succeeded in conferring immunity against tuberculosis by transferring white blood cell between guinea pigs. Then in 1950, the lymphocyte, which is a type of a white blood cell, was identified as the cell type responsible for both cellular as well as humoral immunity. Then Bruce Glick at Ohio State University indicated the existence of two types of lymphocytes. One T lymphocyte, which we call T cells, which is derived from a organ, thymus, and B lymphocyte, B cells, derived from the bursa of Fabricus in birds. So this was also another interesting experiment by Bruce Glick from Ohio State University. He studied the function of the bursa of Fabricus, which is found only in birds, a lymph organ which is found in the cloacal region. This red dot, here we can see this red dot, this bursa of Fabricus is found in this region. So he was studying about the function of this organ in birds. So what he saw, he removed this organ, bursa of Fabricus, from the bird, from the chicken he removed. And then he saw that these bursectomized chickens were susceptible to this salmonella. But in chickens where there was this bursa of Fabricus was present, they were found to make this anti-salmonella antibodies when they were inoculated with this salmonella bacteria. But when he removed it, he saw that no antibodies are found in these birds. So, like this, he found out that bursa was a organ in which antibodies were being produced. So, these antibody producing cells were known as B cells. So, bursa of Fabricus, so B cells he named it. But however, we mammals do not have bursa of Fabricus. So what we have? We have bone marrow. So bursa of Fabricus is identical to our bone marrow, where these B cells are present and they produce these antibodies. So what humoral immunity do? Humoral immunity involves combating pathogens by antibodies, as well as which are produced by B cells and can be found in bodily fluids, and it can be transferred between individuals to produce passive immune protection. Whereas cell mediated immunity involves the work of pathogenic specific T lymphocytes which can act directly to eradicate the infectious agent as well as aiding other cells in their work. So these are the two branches of immunity. So the next question was, how are the foreign systems recognized by our immune system? So in 1890, Jules Bordet observed that there were sheep antisera to the bacteria Vibrio cholerae caused lysis of the bacteria. So as I already told that they found out that the serum contains some components which is responsible for transferring this immunity to healthy people. So here also Jules Bordet observed that when he took antiserum of sheep to the bacteria Vibrio cholerae, it caused lysis of the bacteria. However, when he heated this antisera, it destroyed its bacteriolytic activity. After heating the antisera, he observed that there is no lysis of the bacteria. So, bacteriolytic activity required two different substances. One was the specific antibacterial antibodies which are resistant to the heating process, and another was the heat sensitive component responsible for the lytic activity. So when he heated it at 56 degrees centigrade 30 minutes, it activated that component. So he named this as a complement. Paul Ehrlich in Berlin, he also did the similar experiment, and he did name this as complement, defining that it is the activity of the serum that completes the action of the antibody. So not only antibody but the complements which actually helps the action of this antibody is also responsible for this bacteriolysis. So Bordet won the Nobel Prize in 1919, where he showed that this complement is also another component which is helping in the bacteriolysis. So there are other theories which are known as side chain theory. So the side chain theory dates back to Paul Ehrlich in 1900. He proposed that cells in our blood expressed a variety of receptors. Not only one receptor, but a large number of receptors are present in the cells in our blood, which he called them as side chain receptors. And these side chain receptors can bind to infectious agents and inactivate them. And he proposed that binding of the receptor to an infectious agent was like that between a lock and a key. So when a lock is binding to a key, similarly these receptors present in the cells is binding to any foreign substance which is entering our body. So he suggested that interaction between an infectious agent and a cell-bound receptor would induce the cell to produce and release more receptor with the same specificity or conformation. So he showed that this interaction between a foreign pathogen and a cell-bound receptor will actually make more receptor of the same conformation or specificity. And he termed this as antigen. These foreign particles, he termed this as antigen, which can elicit a specific response by B or T lymphocyte. Here we can see side chain theory. Here we can see one cell with different types of receptors. 1, 2, 3, 4 receptors we can see. These 4 receptors are specific to 4 different pathogens. Here we can see different kinds of pathogens are there. And when these receptors, suppose this black colored pathogen, when it is coming and interacting with one type of receptor, we can see that that same receptor are being emerging in that specialized type of cell where and more amount of that specific receptors are being formed in the cell. And so what he according to him, there are different side chains which are specific to different antigens are nothing but substances which are present on the uh, foreign substances, and these antigens can elicit an immune response. And uh, in his initial theory he told like the cell is pluripotent, means it can generate a number of different receptor or side chain on its surface, and all of these receptors is having different specificity. So if an antigen and encounters this cell and has a good fit with one of its side chains, synthesis of that receptor is triggered and the receptor will be released. So depending on the type of antigen which is entering into our body, that receptor is being made, and then it can interact with that specific antigen. Because we all know that in our body there are different, we are exposed to different kind of pathogens. So no, so our body can recognize different kind of pathogens, be it bacteria, be it virus, be it anything. So that means our body has been like made in such a way so that it can recognize different antigens, and these antigens are also recognized by different receptor. Not one antigen can recognize both virus it can recognize, or both bacteria it can recognize. So there are different types of receptor present for different kinds of antigens. And so whenever our cell comes in contact with one specific antigen, it starts producing more and more of that particular receptor. So this was known as side chain theory by Paul Ehrlich. Another theory given was clonal selection theory. So this clonal selection theory was like an individual B or T lymphocyte. As I already told, there are two types of lymphocyte, B lymphocyte and T lymphocyte. They express many copies of membrane receptor that is specific for a single or distinct antigen. That is, there are different type of receptors which are specific for a particular antigen. So this unique receptor is already determined before it is exposed to the antigen. It is already present, that unique receptor is already present in our cells before we are exposed to a particular antigen. So first, whenever there is a first encounter of this antigen with a receptor, with a specific receptor, it will activate that particular cell, causing it to proliferate into a number of daughter cells that have the same receptor specificity as the parent cell. So clonal means it will produce similar type of cells, clones of cells it will produce after it is getting exposed to a particular antigen. So here we can see that this is a mice, and this had been exposed by a foreign protein or any infectious agents. So there are two types, as I already said, humoral response and there are cell mediated response. So in humoral response, we have our B cells which produce the antibody. So this B cell, suppose, is getting this antigen is coming, and this there are these receptors which are known as B cell receptors present on the B cells. So this antigen is coming and interacting with the B cell receptor. So this is the first time our B cell is being exposed to this antigen. So it will produce antibody against this antigen, and that will remove this pathogen. However, in case of T cell also, there are T cell receptor which are present. This T cell receptor will interact again with this antigen, and it can directly kill these infected cells, or it can produce some molecules which we call cytokines, which will indirectly kill this pathogen. So the humoral response involves interaction of B cells with foreign proteins called antigens, and the differentiation into antibody secreting cells. Whenever there is interaction of the antigen with the B cells, these B cells will differentiate into more and more of antibody secreting cells. And the secreted antibody will bind to this foreign protein or infectious agents and will help them to clear them from the body. And in case of T cell or the cell mediated response, it involves various population of T lymphocytes, and these T lymphocytes can perform many functions including the secretion of, as I already told, the cytokine, the soluble messengers that help directly other cells of the immune system, or it can directly interact and kill the infected cells. So any question is there, because uh, you can follow what I am telling. Any question is there? If any question is there, you can write in the message box also. Whether you can understand what I am telling, or is it difficult to understand? Uh, so no question? No question, and everyone can understand what I am telling? Only Elias has written that understandable. So other people, Aishwarya, Harita, okay, everything is understandable. Okay, shall I proceed further? Okay. So we know that there are different types of pathogens we are exposed every day. Whenever we are walking, like sleeping, eating, we are exposed to a large number of pathogens. And this pathogen can be anything. They come in many forms, and they are like, from their size also varies from small to big. Different kinds of pathogens are there surrounding us every time, and we cannot see these pathogens. Only our immune system can see this when this pathogen is entering into our body. So what are pathogens? So pathogens are nothing but, pathogens are the organisms which are causing disease. We call these organisms causing disease as pathogens. And what is pathogenesis? The process by which they induce any illness in the host is called pathogenesis. So, whenever a virus is coming and entering into our body, that virus we can term as pathogen. And whenever it is causing, any virus is causing any illness in our body, we can call this as viral pathogenesis. The way they induce the illness is known as pathogenesis. So there are mainly four types of pathogens around us. One is virus, one is bacteria, one is fungi, and one is parasite. So we all know, we are all familiar with this kind of uh, pathogens, and there are different types of viruses, there are different types of bacteria, there are different types of fungi, there are different types of parasite. So our body is very much adapted and very much like used to like combat all these kinds of viruses. Not one virus, all the viruses will not cause the same symptoms or same amount of immune response in our body, and all the bacteria also will not cause the same kind of immune response in our body. So our body can fight the different types of viruses, different types of bacteria, different types of fungi, and different types of parasites in different ways. So you can understand how our body is trained to fight all these pathogens we are exposed every day. So our immune response quickly becomes tailored, like it should act quickly to suit the assault, to suit, like to combat the pathogen. It should not be slow, it should act very fast. So during the initial stages of infection, the receptors that first recognize the foreign agent help the immune response categorize the offender and tailor the subsequent immune response. So during the initial stages of infection, there are certain receptors which first recognize this foreign agent and help in combating this infection by activating our immune system. And there are different types of unique pathways that emerge together, and they are specific for different types of pathogens such as cytotoxic T cells that kill viral infected host cells, and T helper cells that assist other immune cells, as well as antibodies secreted by B cells to fight extracellular infection. So there are unique pathways, there are unique receptors which are specific to certain pathogens, and they will start producing different unique, they will start the activity of different pathways, and which will together emerge together to fight off this infection. So most of the pathogens express at least a few chemical structures that are not typically found in mammals. So suppose some bacteria is coming into our body, it expresses certain molecules. Obviously, our body will not have certain structures which is present in the bacteria. So our body can easily identify that some foreign pathogen has entered into our body. There are patterns known as pathogen associated molecular patterns, which are common foreign structures that can characterize whole group of pathogens. So there are certain molecules which are present on these pathogens which our body can easily recognize. These common foreign structures, when our body recognizes, then it can induce our immune response and that can combat the infection. So animals, both invertebrates as well as vertebrates, have evolved to express several type of cell surface markers as well as soluble proteins that quickly recognize many of these pathogen associated molecular patterns, which is a kind of pathogen profiling. So be it human beings or be it also animals, both invertebrates as well as vertebrates, they have evolved to express several types of cell surface and soluble proteins. Like as I told in our case, there are antibodies, there are T lymphocytes, B lymphocytes, which can quickly recognize many of these PAMs, and other cell surface receptors which can act very quickly to combat the infection. Not only human beings, but both invertebrates as well as other vertebrates have evolved such kind of immune system. So there is an ongoing race between host as well as pathogen. So pathogen obviously wants to stay and in our body, but our host system also wants to actually combat the pathogen. So pathogens have evolved to express unique structures that avoid host detection, as well as our host also co-evolves to match these new challenges. So there is an arms race between pathogen as well as between the host. So pathogen wants to circumvent our immune system and we want to remove the pathogen from our body. So both host as well as pathogen is evolving every day to uh, like combat each other. So, as we already know, there are terms we call as resistant bacteria, antibiotic resistant bacteria. Why? Because these bacteria have evolved strategies to resist these antibiotics, and so we term this as antibiotic resistant bacteria. Also, there are many viruses, especially there are viruses which RNA viruses whose genome are made up of RNA. These viruses who actually evolve a lot, they change their structures, they change their properties so that our host system cannot recognize these viruses. So in that way, these pathogens want to avoid host detection. But our host system also knows that these pathogens are constantly evolving, constantly changing. So our host also is co-evolving to match these new challenges to combat the pathogen. So there is a kind of race, there is a kind of fight between the host as well as the pathogen. Because in case of SARS-CoV-2 also we saw that with different time in different time period different variants are coming, so these different variants are like these pathogens are evolving, they are making their new strains to avoid our host detection. So like this way there is a kind of race between the two, host and pathogen. So, how can our immune system be prepared for this? Or how can our DNA encode a recognition system for things that change in random ways over time? And how do we every time build a system to recognize new chemical structures that may arise in the future? Like how we are prepared, like how we also have to get prepared for matching the pathogens. So, how do we do that? So there is a generation of diversity. It is employed by B and T lymphocytes. So as I already told, like there are cellular mediated immunity as well as humoral mediated immunity. So this group of B and T cells in which each cell expresses many copies of one unique recognition molecule, collectively a cell population with a theoretical potential to respond to any antigen that may come along. So here we can see that there is a generation of diversity. This stem cells is producing, this stem cell is the first cell from where our B cells or T cells are coming up. So from this stem cell, you can see different colored cells, yellow, pink, blue and green. So all these cells are producing different kind of receptors. These receptors are uh, like uh, these receptors can recognize different specific kind of antigens. So this yellow can recognize a different antigen, this pink can recognize a different antigen, this blue can recognize a different antigen, this green can recognize another different antigen. So all these four can recognize four different antigens, and suppose this pink receptor as we can see is recognizing a particular antigen, and suppose that antigen is coming and that antigen is coming and other like yellow, blue and green cannot recognize this antigen. So what happens, this pink and this pink cell can interact with this antigen, and then there is clonal selection and expansion, means at that time only this pink cell can be produced in large numbers to combat the antigen. Others won't be produced. So by this, there is like clonal expansion and selection of this particular cell when the particular specific antigen is coming. So, and also this is done by rearranging and editing the genomic DNA that encodes the antigen receptors expressed by B or T lymphocyte. This we will talk in next classes. Like there is editing, like we know that from DNA comes RNA and from RNA comes protein. So in the DNA itself, there is huge combination. Like they will rearrange the DNA, these receptors, the DNA of these receptors will undergo rearranging as well as editing, and this will produce new and new kind of B and T lymphocyte. With permutation and different permutation and combination, this DNA will of this B or T lymphocyte will generate new and new kind of receptors, so that any kind of antigen, if it is coming in our body, can be recognized by our body. So B and T cells make surface receptors unique to each individual, which are then not passed on to the offspring. However, uh, like these are not being passed on to our offspring. They make unique receptors in each individual. Means if I am having some receptors that is unique to me, this will not be present in another individual. So some people are susceptible to some disease while resistant to others, and some, like I might be susceptible to some particular disease, but I might be resistant to some particular disease. Another person is can be susceptible to a particular disease and resistant to other disease. So these receptors are unique to each individual, and it won't be passed from us to our offspring. However, there are certain common elements which can be inherited and passed on to next generation, which we call as pathogen recognition receptor or PRRs. These are present on everyone. These are unique, not unique. These are common in all the individuals, and they are present on the cell surface to recognize the pathogens. So, in contrast to the B and T cells, these are not like changed, or their DNA are not rearranged, but they are inherited and passed on to next generation. So there is a generation of diversity which actually helps us to recognize a diverse number of pathogens. So another thing is tolerance, immune tolerance. There is a, like, we all have some immune tolerance which ensure that our immune system do not destroy our host system. Means if our immune system is hyperactive, then it can destroy our self, means our body it can destroy. But there is certain term known as tolerance that ensure that our immune system is working fine. It is not hyperactivated. So one, ma'am, can we, Ramonjit has sir? Yes, you can get the PDF of this presentation. Till now, everyone can understand everything, or any question is there, you can ask anything. Any doubt is there? Okay, no doubt. Um, so there is a tolerance in our immune system which avoids to destroy the host. So as I already showed that there is a generation of diversity. Diverse receptors, diverse cells are being produced to combat different kind of pathogens. However, there might be some leakage. Leakage means suppose this kind of random, not yet, ma'am. Okay, no doubt. So, like diverse receptors are being produced, as I already told. So this diverse receptor by chance can target the host. Random permutation combination of these receptors. Suppose a particular receptor is being produced which is interacting with our own host protein, suppose, then it can cause damage to our host. So this immune tolerance is very much important, so that our immune system don't attack our own hosts, means attack our cells. So in order for the immune system diversity strategy to work, it must somehow avoid accidentally recognizing and destroying our host tissue. So if this much permutation combination is happening and this much receptors are being produced, then we have to also check that these receptors by chance do not target our host system, host proteins, host molecules. So there, so there were two people, Barnett and Medawar, who were awarded the Nobel Prize in Physiology or Medicine in 1960 for their foundational work characterizing immune tolerance. So, suppose here we can see that it is a self marker. This cell can easily identify that this is our self marker and we should not target it. However, when a pathogen is coming, that cell is like, yes, this is a pathogen and this is not us. This is not our self marker and we should target. So here we can see that the cell is happy and it is not targeting the self marker, and the cell is very angry, like it has to target the pathogen. So there is this immune tolerance should be present in every individual to avoid destroying the host. So they should avoid the self markers and they should identify what are the non-self markers. That is, the self and non-self discrimination is very much important for an immune system to actually work perfectly. So as I already told, like this is a neonatal tolerance demonstrated by an experiment done by Peter Medawar in the 1950s. This what he did was like he took some mice, and after he took that mice, he has uh, took from there is this is a recipient strain A, and uh, after it has become adult, what he did was from a strain B, which is a completely different strain, he took some skin and he put that skin and from the donor strain B grafted it into the recipient strain A. It's just introduced that skin from recipient B to recipient A. However, he saw that this graft was rejected, that mice died. However, what he did in another case, he injected some spleen cells from strain B, and after that he saw that whenever he is grafting the skin of donor B into recipient strain A after the injection of the spleen cells, this graft is accepted. That means in the first case, what is happening? Our immune system is recognizing that this skin from the strain B is not ours. It is not self. It is a non-self. So it is kind of pathogen. According to it, it is coming from outside. So it is a pathogen. So it is rejecting and it is activating the immune system, which is killing that mice. Like, or it is the some immune response are being created, which so the graft is kind of rejected in the first case. But whenever we are giving some spleen cells from the strain B, from spleen cells, if you are giving, then that's in the, however we have to do it in the neonate stage. That is why he has taken neonatal tolerance. When, because in the like early stage only our tolerance system, like we are we can recognize self and non-self in the early stage only. If we are in the adult stage, and adult stage if you are giving, then it can again consider it as a foreign pathogen. So he is giving this spleen cells from the strain B in the neonate or when it is already a baby mice kind of. So when it is giving that and then it is pushing giving that skin from donor B, then it is he saw that the graft is accepted and like the mice is surviving. So this proved that the in our early stage our cells are made to learn that this is our cell, this is pathogen cell, this is self cell, this is pathogen cell. Like And this phenomena is known as self-tolerance, which prohibits immune response to host tissue and is maintained through the elimination or inhibition of cells or receptors that could respond to cell structure. So, suppose some like leaky system is something has like some portion of our recognition receptors or the B or T lymphocyte has accidentally been produced, which is recognizing our own cell. So our immune system will quickly try to remove or destroy that cells which is responding to our cell structures. So these phenomena are being there in our host cell. So you have to have a minimum amount of self tolerance to avoid like activating the immune system reacting to our own host tissue. So another model was proposed by Polly Matzinger. She told like immune system constantly evaluates each new encounter more for its potential to be dangerous versus safe to the host than for whether it is self versus non-self. That is, it has the ability to distinguish between good death or bad death. By good death, what we mean by good death? So, good death is every day normally in our biological events, we need a cleanup response to remove some debris, to remove any dying cell, to remove anything like which is not required anymore. Any kind of uh, like cells or debris or our cells after aging of the cells, these cells needs to be removed. However, bad death is what are the warning signs that include the release of intracellular contents, expression of cellular stress proteins, and sometimes also pathogen specific products. So bad death means suppose a pathogen is coming, that pathogen is affecting a particular cell, that particular cell is getting is dying, it is on the verge of death. So it is not a natural phenomenon, it is due to the pathogen that that cell is dying. So that is a bad death. But suppose our some particular cell is aging, it has aged and it is not required in our body anymore. So our body wants to clean that. So that is a good death. That is a everyday biological process which is happening. So our body should able to distinguish between this good death and this bad death. So she proposed the danger or the damage model of self-tolerance, which shows that the immune response is not activated when host cell death occurs safely, but when only this immune response is activated when this death is accompanied by other damage associated signals produced by the host cells. So it should be a good death and it should be a bad death for our immune system to get activated and not a good death. So their self and non-self discrimination should be there by our immune system. So uh, any question? Any question till now? So what are the two different branches of immunity? Anyone can tell? How does the immune system play a role in death of the cell? Okay. Okay, like I am telling that there can be, suppose in our everyday process, many cells are, we don't need many cells. Sometimes there is normal cleanup process. Our body is normally doing a cleanup process. Like this cell is not needed, this cell is has age, so we do not need this. So we need to clean this cell. So this is a good test. Means our body is only cleaning up this. So, but there are certain deaths which are bad death, which are warning signs, like some pathogen is coming in our body. That particular pathogen is making a cell die. Suppose a healthy cell is there, that pathogen is coming and is destroying that cell. It is releasing some content so that that cell is dying. That is a bad death because that is not happening naturally. That is induced by a pathogen. So, our immune system should recognize that this is a good death, this is a bad death. So, the bad death is caused by a pathogen or maybe due to expression of other cells. Suppose you are in very much stress. So, in that case, that there will be like the cells will die normal, not normally, but due to stress, there will be death of the cells or death of any uh, particular cells which can produce any kind of patterns which is showing that this is a warning sign. So our immune system has to recognize that this is a bad death, this is not a good death, this is not a regular cleanup process that is happening every day. So our immune system should recognize these warning signs which are caused by the pathogens or due to any stress, that this is a bad death and we have to activate our immune system. But whenever there is some good death, like normally cells are dying in a cleanup response or any metabolic process, we need to remove some extra things from our body, then our immune system should not be activated. If our immune system is activated whenever there is a normal cleanup process, then it can damage our tissues. Damage because it is not normal. It is not normal for our immune system to get activated by normal physiological processes. But whenever there is a pathogen which is coming, then it should get activated. It should not get activated when there are normally physiological processes happening in our body. So this only this Polly Madzinger, she told it as a model or a danger or damage model. So whenever there is a bad death, our immune system should get activated, and whenever there is a good death, there should not be any activation by the immune system. So you understood? Okay. So there are two branches of immunity. I was already told based on the cell types. One is humoral immunity, and another is the cellular immunity, as I already told. Apart from that, there are our immune system is composed of two interconnected arms. One is innate immunity and one is adaptive immunity. So innate immunity and adaptive immunity. Can anyone tell the difference? I think Sir has also taught what is innate immunity and what is adaptive immunity. Anyone? Adaptive and innate? What are the differences between adaptive immunity and what are the and innate immunity? Innate immunity which is not get affected by the which is not get affected by the pre-exposure of the pathogens. Okay. Anyone else? Uh, innate immunity is non-specific while adaptive is specific. Yes. Innate is the natural immunity, I mean. Okay, okay, understood. Yes. So, innate immunity is innate immunity is present in us since the time of birth, but adaptive, we as days passes, we gain this adaptive immunity. So the response time, we can see here a table which can show the difference. Like the response time of innate immunity is my minutes to hours, while adaptive immunity can take days to develop. Specificity of innate immunity is limited and fixed. Adaptive immunity is highly diverse, adapts to improve during the course of immune response. Response to repeat infection is same every time in innate immunity. However, in adaptive immunity, it is much more rapid and effective with every subsequent exposure. Major components of innate immunity are the phagocytes, the barriers, the example skin, pattern recognition molecules, while in adaptive it is antibodies, T cells, B cells, and antigen specific receptors. So, innate, as I already told, innate responses are rapid but less pathogen specific and present since birth using inherited recognition molecules and phagocytic cells. Adaptive responses are slower, very much taking days to develop, but highly specialized. It is very specialized and can rely on randomly, as I already told, some random generation of the T cells and the B cells. And both of these immune response are two interconnected arms. They operate cooperatively. It is not that only in one time innate is acting, then adaptive not acting, then adaptive acting, innate not acting. So activation of the innate immune response produce signals that are required to stimulate and direct the behavior of the subsequent adaptive immune pathway. So obviously innate immunity acts very much quickly. So it is the first line of defense. However, it also activates the adaptive immunity, which is the second line of defense, but it is more specific. But however, it takes days to develop. So both of these are the two interconnected arms. One cannot only, it is not that one pathogen is coming, only innate is acting. Another pathogen coming is only adaptive is acting. So everything is interlinked, both the immune responses. So this is our immune cells and molecules which are found in many places. Like immune system, we know, as I told, skin is the different type of physiological barriers which are present in our body, which is having that immune cells. And this is bone marrow where the B lymphocytes, I told, like B lymphocytes and T lymphocytes develop. And this is thymus where the maturation of these T lymphocytes takes place. And these are the lymph nodes. You can see there are different lymph nodes. This is the spleen. I already told in one experiment they have taken the spleen cells and have transmitted into the mice, and there we can see that it can recognize, it got grafted with another strain. So these are the tonsils. We know here we have tonsils. Sometimes due to infection these tonsils get, like, these tonsils will uh, get flared up. So there is mucosa from where also pathogen can enter through skin. These are the different barriers which can allow the entry of the pathogens, and these are the different kinds of immune cells which are present. These are the white blood cells, as I already told, the phagocytes, and everything is present here in the bone marrow. So this is our whole immune system. And here we can see how the whole immune system is acting when a infection is occurring. Like through mouth, you can see this. Suppose the COVID infection, it was through sneezing and through coughing, it was going from one person to another. So as this is the cell barrier through which this infection is entering, or it can enter through any cut or splinter also. Through gut also it can enter through food. Food is a major cause, one of the biggest cause from where we are getting many kinds of infection. So this infection or the bacteria is entering, and here you can see this innate immunity. It is activating the innate immune branch of our immune system. These cells are there which are going and phagocytosing these pathogens. Then this is entering into the lymph node where there are this lymph node is coming, this cells are coming in contact with this B cell and T cell, where this clonal expansion is what like happening. As I already told, this B cell is getting divided and clonally expanded, T cells getting clonally expanded, and more amount of B cell T cells are produced, and from there these lymphocytes are coming out into the, here we can see the heart, and there is like this blood is carrying all these T cells and B cells, where it is giving some effector responses. Here also we can see from the bone marrow the B cells are coming, and from the thymus the T cells and the both the T cells are coming and entering into this lymph organ. So this is how this circulation of the B cells, T cells, and the exposure to antigen is helping in activating the immune system. So this we will be studying later in much more great details. So, as I already told, adaptive immune response typically generate a memory. So, once you are exposed to a particular pathogen, suppose you are infected by a COVID-19, so that our immune system will remember that we are infected by this COVID virus, and then it will again when similarly again when virus is coming for the second time, it will again uh, like generate some, like it will have a memory like, yes, I have been exposed to this virus, so it will generate a response very quickly compared to the first time. So here we can see a graph, like this is a primary response. Antigen A is coming in the 0th day. There is a primary response. So when an animal is injected with an antigen, it produces a primary antibody response. So antibody will take time to develop, of low magnitude and short duration, peaking at about 10 to 20 days. So it is peaking at about 10 to 20 days. This antibody production takes time. However, if we are getting exposed to the same antigen in a secondary at a later time point in a secondary response, that peak is in less time, that is 1 to 4 days it will take, and it is more antigen specific than the primary response. Here you can see again we are getting exposed to the antigen A, and which is actually this response is much more compared to the previous response. So our immune system remembers that there is a previous exposure of antigen A, and then it quickly like respond to the second respond quickly when there is a second exposure of the antigen A. So adaptive immune response typically generate memory. However, you can see innate immune response which have no memory element. It do not have any memory element, and it occurs every time an antigen is exposure. Means the same antigen A, even if it is occurring in the second time also, it will be the frequency of it acting will be similar to the previous exposure. However, in case of adaptive immunity, it is much more heightened. The immune response is much more heightened and effective. But and it will also take less time compared to the first time. So we are already only talking about the good sides of the immune system, but however, there can be inappropriate or dysfunctional immune responses which can result in a number of disorders, and most instances of immune dysfunction occur in three broad categories. One is hypersensitivity, one is immune deficiency, one is immune imbalance. So we all know that hypersensitivity, or some of us have some allergy to some things like pollen, some dust. And why this is happening? This is because uh, our immune system is attacking the self protein or tissues by the immune self proteins or the tissues. Like, instead of the foreign antigen, they are attacking our own system, so that can cause to hypersensitivity. There can be immune deficiency, like our immune system is not that much active in response to any foreign antigen, so there is a deficient in our immune system which can cause an insufficiency of the immune response. And there can be immune imbalance, like dysregulation of the immune system can lead to aberrant activity of the immune cells, which can increase inflammation and or reduce immune inhibition. As I already told, like, not it should not be excessively activated, and it should not be like very, it should be balanced. Our immune system should be balanced. It should not go like it is too much activated or too much dysregulated, then it can cause some inflammation or some different types of disease. We will again learn this in the next classes, what are the different kinds of diseases. And then another is hygiene hypothesis, which was given by D.P. Strachan. So he told, like, there is a decrease in human exposure to previously common environmental microbes has adverse effects on the human immune system. So the more you are exposed to our environmental microbes when you were a child, the more your good immune system will be. So more you are like having a very good hygiene when you are like a child, then more you are prone to become sick. So several categories of disorders caused by excessive immune activation have more been prevalent in industrialized nation due to diminished exposure to particular classes of microbes following the widespread use of antibiotics and overall hygienic practice. The more you are exposed to the microbes from the early stage, the more you have a good immune system. But it is seen that more industrialized nation, like very much industrialized, or there is very much cleaner place is there, or you are very much exposed to antibiotics or over on hygienic practices, then in those particular places there will be like the immune system won't be that much good, because in the later period of time, whenever you are getting some exposure to antigen, there will be excessive immune activation which can damage your tissues. So childhood exposure to cow sheds, farm animals, even having several older siblings, then attending daycare early in life, growing up in a developing nation has all been related with a decreased likelihood of developing allergies. So this was the hygiene hypothesis given by D.P. Stratton. So that's all for today. So if you are having any question, please ask me. Any doubt you are having, you can ask me. Any doubt regarding whatever I presented today, or any doubt like mm, inserts course in first week, any doubt with the assignment, you can ask me. Any kind of problem you are having in understanding. No doubt, no questions. Okay, I did not complete. Okay, okay, so no one has any doubt. If anyone has any doubt, you can also mail me. And regarding today's lecture, if anyone is having any doubt, or you can ask any question, because this is a interactive session, so need not be like I only has to speak. You can also ask any kind of question you want. So
Hello, ma'am.
everyone, yes, yes.
Good evening, ma'am.
Good evening.
मैम क्या आप मेल शेयर कर सकते हो आई मीन पीडीएफ इफ यू कैन शेयर इट
Uh, I can share.
ओके
Uh, you can like write the mail ID. I can share.
ओके मैम प्लीज
No one has any doubt. Thank you. Okay. Thank you. So thank you everyone for listening. And if you are having any doubt, I have written my mail ID in the message, so you can ask me. You can even write in the PMRF that course portal also if anyone is having any doubt. So we can like answer your queries. No questions? Then thank you everyone. We will meet again next week, Tuesday. Even if you want me to talk about any particular topic, also I can teach you. If you are interested in any particular topic, also I can teach you. If you want, you can write in the message. Then I will make the slides accordingly. And if you are having any interest in any other topics, also you can tell. And please go through the NPTEL course. And any doubt is there, you can mail me. So
Thank you.
thank you everyone. Then we can stop here. And even if you want anything, you can just mail me, or in the portal you can write anything if you're having any doubt. Thank you.